Bom, boa tarde. Voltando agora para mais um período de palestras. Agora à tarde nós teremos mais 4 aulas. Eu espero que vocês estejam aproveitando o evento. Obrigada pela participação de vocês. Sempre que existe uma interação, a discussão fica mais rica e o conhecimento aumenta, né? O evento ganha uma outra dimensão. Então, vamos lá. Vamos começar. Eu vou vou retomar com uma apresentação dupla, que será da professora Emily Lavoie e do aluno Charles Hodgman, com a aula intitulada Metabolismo energético, Leucócitos e Exercício físico. Charles atualmente trabalha no Departamento de Saúde e Desempenho Humano da Universidade de Houston, no Texas, e faz pesquisas em fisiologia do exercício, imunobiologia e biologia humana. E está finalizando seu doutoramento sobre orientação da doutora Lavoie. Já a professora Lavoie é a professora associada da Universidade de Houston. Os seus estudos tentam entender como a aptidão física e os exercícios impactam no sistema imunológico, e especialmente entender como a aptidão física e os exercícios podem melhorar doenças e condições associadas à desregulação imunológica, como o câncer, por exemplo. Ela também é interessada em como a aptidão física e o exercício podem mitigar o impacto do estresse no sistema imunológico, utilizando os modelos humanos e animais. Atualmente, ela é membro do American College of Sports Medicine, da American Physiological Society e da International Society of Exercise and Immunology, da Psychoneuroimmunology Research Society. Dr. Lavoie and Charles, thank you for joining us today. It's a pleasure to have with us, and the floor is yours.
Thank you very much. Thank you for the introduction. I'll just go ahead and let's see if I can share the PowerPoint. I'm going to do that. And then, okay, hopefully this works.
We'll see.
There we go. Are you seeing? Oh, wait, there we go. I believe you should see that.
Yeah, now you'll see.
Okay, get the right window up.
I think you should start with the presentation mode.
Yeah, it's the button is evading me at the moment. Here we go.
Yeah.
There we go. All right.
Perfect.
Out of the way.
It works.
As I've been introduced, my name is Charles Hodgman. I'm currently a PhD candidate under Dr. Emily Lavoie at the University of Houston. I want to thank the organizers for inviting myself and Dr. Lavoie to speak with you today. Immunometabolism and exercise are keen interests of our group, and we're excited to share some of our research in this area. My presentation will proceed as outlined to the right. I'll begin with a brief background as to the rationale behind our current project, as well as a description of the study itself. I'll then present some preliminary results from the data we've thus far collected before before passing on to Dr. Emily Lavoie for her presentation. I need to emphasize that the results I'm sharing are preliminary and we have yet to publish them, so please refrain from copying, sharing, or otherwise saving those data. Broadly, our group does research in the area of exercise immunology, the effects of exercise on the immune system. As I assume many of you will be familiar, much has been done in the field to describe what exercise does to the immune system. That is the manner in which the immune system responds to acute and chronic exercise. Building on this work, I believe the relevant question now becomes how exercise affects the immune system. That is, what are the mechanisms by which acute and chronic exercise alter the immune system? Our group suspects that cellular metabolism is a prime vector through which the effects of exercise are communicated, and this has informed our current research. To be more specific, the following observations led us to the current project. I understand this may be a review for many of you. Please excuse the repetition, but I feel the background is useful to help set up the rationale for the project. First of all, it's well established that acute exercise leads to an increase in metabolic activity, and that metabolic activity and substrate utilization are dependent on exercise intensity. High intensities necessitate greater energy turnover and increasing proportional use of carbohydrate versus fat. While such responses primarily occur within the skeletal muscle, there are coordinated effects throughout the body in order to supply the muscles, effects to which leukocytes are likely sensitive. Second of all, the field of exercise immunology has established that acute exercise leads to a mobilization of leukocytes into the peripheral circulation, also in an intensity-dependent manner, higher intensities of exercise, leading to a greater mobilization of cells relative to rest. Furthermore, among leukocytes, lymphocytes, such as CD8 T cells, are preferentially mobilized. And among lymphocytes, antigen experience effector subsets of those cell types are proportionally mobilized to a greater degree, as can be seen in the figure at the center of the screen. The ascending bars reflect the proportionally greater mobilization of more differentiated CD8 T cells relative to their resting levels, post being immediately post-exercise. Thirdly, acute exercise may also affect leukocyte function. While it may occur that exhaustive exercise leads to a transient suppression of immune function, generally, acute exercise appears to elicit no change or an increase in leukocyte effector function, also in an intensity-dependent manner. This is evidenced in the figure currently at the center of the screen with the elevated levels of interferon gamma positive CD8 T cells in response to greater intensities of exercise. In this case, 95%, 105%, and 115% of lactate threshold. Finally, work in the greater field of immunology has demonstrated that cellular metabolism is fundamental to leukocyte form and function. CD8 T cells exemplify these patterns. In general, naive and unactivated cells exhibit lower metabolic activity and utilize a mixture of glycolysis and mitochondrial respiration to meet those needs. Upon activation, effector cells increase metabolic activity and shift their bias towards the oxidation of glucose via glycolysis to provide energy and biosynthetic precursors for proliferation. Following the resolution of infection, memory cells are those that have retained greater bias to mitochondrial respiration, and these cells rely on mitochondrial respiration of fats and glucose to maintain viability for future pathogen encounters. Collectively, these findings reveal the profound and intensity-dependent effects of acute exercise on energy metabolism and the immune system, as well as the striking similarities between metabolic shifts that occur with exercise and in leukocytes with activation. The logical questions, at least from our standpoint, are then, how does exercise affect CD8 T cell metabolism? Are the effects intensity-dependent, and are the effects subset-dependent? These are the specific questions that informed our current study that I'll now describe. Our current study is a randomized crossover trial including 3 exercise visits, 1 for enrollment in VO2 peak testing and 2 submaximal exercise bouts. Participants in the study are healthy adults 20 to 40 years old capable of exercise on a cycle ergometer. All eligible participants complete a VO2 peak test on a cycle ergometer. Only those that achieve a VO2 peak of 28 to 42 milliliters of oxygen per kilogram body weight per minute are invited back for the main experimental visits, visits 2 and visits 3. This range of fitness was chosen to reflect the average range of fitness for the population accessible to us, as opposed to the highly fit or the unfit, and support the translational impact of the study's findings. Visits 2 and 3 each include 30 minutes of cycling at a prescribed intensity with blood samples obtained prior to exercise, immediately post-exercise, and 2 hours post-exercise. The only difference between the 2 visits is the intensity of the exercise. One bout is conducted at 90% of the participants' first ventilatory threshold within the moderate exercise domain, and one bout is conducted at 110% of their first ventilatory threshold in the heavy exercise domain. The order in which participants experience either the moderate or heavy bout in visit 2 or visit 3 is randomized. The VO2 and heart rate associated with these intensities, 90% of VT1 and 110% of VT1, are determined from their VO2 peak test results and inform the manipulation of cycling resistance to ensure the prescribed intensity is enforced throughout each bout. Peripheral blood mononuclear cells are isolated from each blood sample and then processed for assessment of metabolic regulation, in this case, gene expression, metabolic function, and cell function. I will be presenting some preliminary results from the metabolic function data today. Finally, tying back to the questions that inform the study, 30 minutes of cycling is the acute exercise we're implementing. Intensity effects are being addressed through assessment of our outcome measures within overall CD8 T cells, Intensity effects are being addressed through comparison of the exercise in the moderate versus heavy intensity domains. And subset effects are being addressed through the assessment of our outcome measures within overall CD8 T cells, as well as in naive, central memory, effector memory re-expressing CD45RA, as well as CD57 positive T cells. The exercise intensities were chosen such that each bout would occur in a distinct domain of exercise intensity. Physiologically, 3 exercise intensity domains have been defined: the moderate, heavy, and severe domains. Critical power or critical speed defines the border between the severe and heavy domains, and the first ventilatory threshold or the first lactate threshold defines the border between the moderate and heavy domains. Therefore, exercise at 90% of the first ventilatory threshold places participants in the moderate domain, while exercise at 110% of their first ventilatory threshold puts them in the heavy domain. Exercise in each of these domains elicits characteristic metabolic responses that likely underlie the physiological adaptations to training in the domain. Thus, if leukocytes are also sensitive to the specific milieu elicited by a given intensity of exercise, prescribing exercise in 2 different domains should provoke unique responses within the cells. As previously mentioned, another aim of the study is to assess whether immunometabolic effects of acute exercise differ between CD8 T cell subsets. The subsets we selected for comparison are displayed on this slide, along with markers used to define them for analysis. The first 4 subsets represent a sequence in differentiation from left to right, such that EMERA cells are the most differentiated of the 4, possess the greatest effector functionality, and are proportionally mobilized to the greatest extent with exercise. We also evaluated CD57 positive cells among the total CD8 T cell pool. These cells are composed of largely effector memory and EMERA cells, and previous work in our lab has shown that they are preferentially mobilized in response to exercise, even within the latter subsets, those subsets being effector memory and EMERA. It should also be expected that subsets possess metabolic differences related to their functional differences. The lesser differentiated subsets exhibiting lower metabolic activity and relying to a greater extent on mitochondrial respiration and the more differentiated subsets exhibiting greater metabolic activity and being biased towards glycolysis. Before I proceed to the results, I must introduce the technique we're using to assess metabolic function. It's called CNITH and is a relatively new technique to assess cellular metabolism by flow cytometry. An advantage of the technique over other assessments of cellular metabolism is that it facilitates the acquisition of subset-level data without the need to physically isolate the cells from the greater population. Thus, the technique is suited to support the aims of the study. At its core, CNITH relies on a strong relationship between cellular ATP production and protein production, such that protein production may serve as a proxy for ATP production. In sequence, cells undergo a brief incubation with inhibitors to glycolysis, in this case 2-deoxyglucose, oxidative phosphorylation, in this case oligomycin, both inhibitors, or vehicle control. This is followed by another short incubation with the antibiotic puromycin. Puromycin gets incorporated into nascent proteins to facilitate the assessment of protein production using an anti-puromycin antibody. The cells are then processed for intracellular flow cytometry prior to data acquisition. The magnitude of puromycin fluorescence in each condition facilitates determination of 5 outcomes related to cellular metabolism. Fluorescence in the control condition provides a measure of metabolic activity within each population of interest. Relative glucose dependence can be calculated based upon the decrease in fluorescence with the addition of 2-deoxyglucose versus control. Mitochondrial dependence is given from the decrease in fluorescence with the addition of oligomycin versus control. And glycolytic capacity reflects the abilities of the cells to maintain protein and ATP production with the addition of oligomycin when oxidative phosphorylation is compromised. This is the remaining fluorescence in the oligomycin condition. Finally, fatty acid and amino acid oxidative capacity reflects the ability of the cells to maintain energy production with the addition of 2-deoxyglucose when glycolysis is compromised. Altogether, these outcomes facilitate the determination of the level of metabolic activity as well as the metabolic pathway preferences in the cell populations under investigation, examples of which I'll now provide. I must reiterate that the following results are preliminary and unpublished from the study, so please do not copy, share, or otherwise save these data. Though we're still collecting data, 16 participants have thus far completed the whole study, and the characteristics of this group are listed in the left-hand table. The figure on the right illustrates the average relative oxygen consumption, as well as the heart rate responses to each intensity. The moderate bout elicited a response of approximately 47% VO2 peak and 65% heart rate peak, while the heavy bout prompted responses of approximately 60% VO2 peak and 75% heart rate peak. These values are significantly different from one another, indicating or confirming that the relative intensity of each bout was significantly different from the other. Onto the data I imagine you may be more eager to see. Within total CD8 T cells across intensities, we've observed a significant effect of time, that is exercise, such that cells obtained post-exercise demonstrate lower metabolic activity versus pre-exercise or 2 hours post-exercise. I also must note that due to variation in our instruments acquisition of the pure myosin fluorescence, all metabolic activity data, so this particular outcome measure, are relative to CD8 T cell pre for the given day. Thus, pre equals 1 in total CD8 T cells, as is evidenced in this particular figure. In contrast to the significant effect of exercise or time, we have not observed a significant effect of intensity. The response to exercise appears largely the same with each intensity. Considering now the subset level data as opposed to total CD8 T cells, we observe a significant effect of time similar to that observed in total CD8 T cells, as well as a significant effect of subset. Across subsets, post-exercise cells demonstrate lower metabolic activity versus pre and 2 hours post-exercise. And across time points, all subsets demonstrate significantly different metabolic activity except the EMRA versus the CD57 positive cells. Considering now both time and subset together, we've observed significant main effects for both, but no interaction effect. Within each subset, cells from each time point differ from one another, and each subset differs from one another in their overall metabolic activity. However, there are not significant differences in the manner with which the subsets respond to exercise. That is, there is no interaction effect, nor did exercise intensity better explain the variability in the results. Returning back to total CD8 T cells, we observed a significant effect of time in mitochondrial dependence and glycolytic capacity, such that post-exercise cells demonstrated significantly greater mitochondrial dependence and lower glycolytic capacity than 2 hours post-exercise cells, and tended towards being different from pre-exercise. A significant effect of intensity was not observed in these outcomes. In contrast to mitochondrial dependence and glycolytic capacity, we did not observe significant effects of time or intensity in regard to glucose dependence and in fatty acid and amino acid oxidative capacity. As of now, I have yet to analyze the metabolic pathway preference data at the level of the subsets, so you'll have to stay tuned for those particular results. Now provide a brief summary of what I've shared. So the information that informed the pursuit of the current study include previous data demonstrating that exercise exerts significant effects on energy metabolism and the immune system in an intensity-dependent manner. Effector lymphocytes, such as CD8 T cells, are disproportionately mobilized with exercise, also related to exercise intensity and cell subset. And cellular metabolism is fundamental to leukocyte type and function, and CD8 T cells exemplify these patterns. This led us to the question of whether and how acute exercise affects CD8 T cell metabolism. Our preliminary results indicate that acute aerobic exercise leads to a transient depression in metabolic activity and a shift towards mitochondrial respiration, with full recovery, if not overcompensation, following 2 hours of rest. Exercise in the moderate intensity domain versus the heavy intensity domain did not elicit differences in different metabolic responses within the cells, at least at the specific intensities tested. And finally, those CD8 T cell subsets exhibit differences in their metabolic activity. Within each subset, the pattern in response to exercise appears the same. That concludes my presentation, and I thank you for your time and your attention.